Today our focus will be on the large and small intestine, and this will end the digestive organs, and we'll move on to accessory organs next. So far we focused on the food moving through the mouth, the pharynx, the esophagus, into the stomach through the cardioesophageal sphincter, mixing in the stomach, and leaving the stomach through the pyloric sphincter. And so now after the food, or chyme as we called it in the last PowerPoint, we're moving into the small intestine. So the food is metered into the small intestine, small portions at a time, and the pyloric sphincter is what allows that to happen. So the small intestine, once it enters here, the small intestine is rather long. It's 3 meters or 10 feet long. And in this part, we have 90% of the nutrients being absorbed. It's divided into 3 sections. The duodenum is the first section that the chyme would enter. And after that is the jejunum, and then the ileum. So the jejunum is the middle part of the small intestine, and then that final part that attaches onto the large intestine is called the ileum. There is a sphincter at the end of the ileum called the ileocecal valve that would allow whatever is left after the small intestine absorbs the nutrients will move through that sphincter or valve into the large intestines. You may remember me talking about the mesentery, and the mesentery, it's what holds the small intestine to the body cavity. So the mesentery is going to be holding this small intestine in place, and it also holds the small intestines together. So you can't really take the small intestines out and stretch them all out into one long tube without cutting mesentery, because the mesentery is holding the small intestine together as well as holding it to the body cavity. So there's not much movement for the small intestine. And here's just basically what I just said. It extends from the pyloric sphincter to the ileocecal valve. It's suspended from the posterior abdominal wall by the mesentery, and posterior means the back side. So it's going to be attached to the back of the abdominal wall and held in place by the mesentery. And like I said before, it can only process small amounts of food at a time, and that's why the pyloric sphincter only allows a small portion of chyme to enter into the duodenum at one time. So chemical digestion is actually started and completed in the small intestine. There's lots of enzymes that are produced in the pancreas. There's also bile produced in the liver that enters into the small intestine in the duodenum, and that is where the chemical digestion is going to start to work and break down the larger macromolecules that have come in from the stomach. And this is where we are going to see most of that chemical digestion take place. On the next slide, I'm going to show you some of the pieces of the accessory organs that flow into the duodenum and allow for this chemical digestion to begin. So pancreatic ducts will carry enzymes, and the bile duct will carry bile to the duodenum and allow for chemical digestion to begin. Now you don't have this diagram to label yet because these are the accessory organs, but I just want to make you aware of some of the things that are entering into the duodenum. And the duodenum, you can see, is this area right here, this small little portion of the small intestine. And this part right here was connected to the stomach, and this part is connected to that middle part of the small intestine called the jejunum. So we have the duodenum where chemical digestion is going to begin, and so we have some things that are entering into the duodenum from this small little duct that comes from the pancreas and also comes from the liver. And you can see here the bile duct is coming down this way, the pancreatic duct is coming this way. It's meeting, and it's allowing all of those enzymes to enter into the duodenum and start that breakdown of those larger macromolecules that came in through the stomach. So I just wanted to give you a quick little look at why this is occurring here and where these ducts are coming from. Now again, these accessory organs are something that we'll talk about next, so don't be worried about this diagram because you don't have this yet to label. Now we talked about those 4 layers of the tissues that make up the digestive tract, and there's some modifications in the small intestine that will increase surface area. So there's 3 things that I wanted to talk about. The first thing is circular folds. I know that's last on this slide, but circular folds, or plicae circularis, if I'm saying that correctly, these are deep folds that occur in the mucosa and submucosa. So inside, where the food or chyme is actually moving through, we have all of these circular curves, like so, that kind of, I'm not very good at drawing, but kind of go around the whole interior of that lumen, okay? So if I were to continue to draw, we would have all of these circular folds going through the small intestine. So this is like we took a cross-section of the small intestine and you're looking down into it. On the circular folds are all of the villi. Now villi are really small, and I'll put those in a different color, and hopefully I can, they're like these little finger-like projections that come off of the circular folds, like so, okay? And they're increasing surface area. So circular folds are increasing surface area, and then all of those villi that are coming off of the circular folds are also increasing surface area. This all helps with absorption of the nutrients. The villi are filled with capillaries and lacteals, and they are what are going to absorb all of the nutrients that your body has eaten. On the villi are microvilli. So if I were to take a villi, and I'll just draw one, okay, like so. On the villi are all of these finger-like projections. It's almost like a toothbrush, and they're called brush border or microvilli, and there's enzymes that are released from that brush border that also help break down any of those larger macromolecules into smaller molecules that our bodies are able to absorb. So these 3 things are modifications to the small intestines. And so as the food is entering into the small intestine, it is very easily absorbed because we have so much of this excess surface area, and that's why 90% of your nutrients are absorbed here. Now you do have this diagram in your packet, in your definition packet or definition worksheet. It's at the bottom right next to Peyer's patches. It's a smaller little diagram, and there's some things there that you should definitely want to label. Again, the lumen, that's the opening. You can see here the circular folds. This is what I was telling you about. It's all of this area right here that I'm doing kind of outlining in green. Those are all the circular folds. So we didn't see that in the esophagus. We didn't see that in the stomach, but we definitely see those extra folds in the small intestines. The folds that we saw in the stomach, we did see folds there, the rugae. Those folds, though, are just for expansion. That's the only thing there. These folds are not for expansion. These folds are to give more surface area to the small intestines to allow for more absorption. So let me erase what I did there so we don't get confused. And then, so you want to label circular folds or plicae circularis. On those circular folds, you can see all that kind of fuzz that's there. Those are your villi, those finger-like projections that are coming off of the circular folds. And then you can see we're back to 2 muscle layers. The inner and outer muscles. Stomach is the only place where there's 3 muscle layers, that extra oblique layer. So we don't see that oblique layer happening in the small intestine. The other thing, if you want to label, are those blood vessels I was talking about. These serve the small intestine. Some of them are bringing oxygen to the cells there. Some of them are absorbing the nutrients and taking them back to the liver or other parts of your body. And those blood vessels are coming up into the villi, so those you're going to see down here in the circular folds. These blood vessels up here that I'm having you label are bringing the oxygen and supplying the cells there with oxygen. Now I don't have this one for you to label, but this is kind of just taking part of the circular folds and enlarging it to show you the villi and what's in the villi. So each one of these projections here is a villi or villus. And you can see that they're lined with absorptive cells. Those cells are what are going to be pulling nutrients out of the chyme that is coming through the small intestine. You can also see that there's lacteals and blood capillaries that are filling the villi. The lacteals are green, the blue and red are the blood capillaries, and they are what are going to take from the absorptive cells. They're going to take those nutrients back. Most nutrients are going to filter back to the liver and then be taken out into the body, and some will just go out into the body. There's some other things we're not really too worried about in this. I just wanted you to see the cells, the lacteals, the capillaries, and the villi. And notice that the submucosa is part of this as well. So we have submucosa and then the mucosa where the absorptive cells are on the villi. So I just wanted to point those things out in a more expanded look at that part of the small intestine. And here I'm taking the villi and showing you the microvilli. So each one of these, whoops, forgot to hit my drawing tool. Each one of these areas right here is a cell that is on the villi. So it's those absorptive cells, and then on those are all of these more finger-like projections that I like to call brush border, and that look to me like a toothbrush. If you were to brush your teeth, you would be brushing those across something like your teeth, and they would be cleaning, in this case absorbing, helping to absorb. Little enzymes are excreted there to also break down any macromolecules that still need breaking down, and those are called brush border enzymes. Now there's a couple things I want to mention before we move on to the large intestine. I didn't mention that as you move along the small intestine, the modifications, the circular folds, the villi, the microvilli, they all decrease in number as you get towards the end of the small intestine. By the time that chyme gets to the end of the small intestine, you have absorbed almost everything you're going to absorb. So as you get to the end, there's less and less of those modifications. And there's also lymphatic tissue that I want to mention. It's called Peyer's patches. It's number 8 on your worksheet. And those Peyer patches are found in the submucosa towards the end of the small intestine, because sometimes the food residue that is pushed through the small intestine may have bacteria in it. And so it monitors that bacteria population and prevents overgrowth of bacteria. Some of those bacteria are good bacteria that we need in our digestive health, but some, they don't want there to be an overgrowth of that bacteria. So Peyer patches is going to be taking care of keeping the bacteria populations under control. I don't have that on the slideshow, but it is a definition that comes up on your definition worksheet, and it's number 8 on there. So now your food is going to pass through the ileum of the small intestine, and it's going to go through the ileocecal valve or sphincter to move into the large intestine. The large intestine is not longer than the small intestine, it's just wider. That's why it's called the large intestine. So the diameter of the small intestine is about 2.5 centimeters. The large intestine, it's about 6.5 centimeters in diameter. It is only 5 feet long, so it's about half the size or length of the small intestine. The small intestine was about 10 feet long. It's made up of longitudinal muscle fibers, it has longitudinal muscle fibers on the surface, and these are called Tenei coli. And let me just show you where they are. It's this muscle that runs along the long intestine. And what it does, it's almost like a piece of elastic that maybe if you were wearing a pair of leggings or sweatpants, you have that elastic that is at the top, and it kind of puckers in the waist of your pants. Well, it does the same thing to the large intestine, and it kind of pulls the large intestine in, and it gives it a puckered look, and it makes all of these haustrum are what these called. These little pouches of the large intestine. So the wall of the large intestine puckers, and it's called haustra, or each one individually is called haustrum. And that term is right here on this diagram. And again, you've got the same tissue layers. You have the mucosa, the submucosa, you have a muscle layer. Again, there's only 2 muscle layers in the large intestine, inner and outer circular muscles. So those are there as well. And so that's some of the diagramming I want to do here as far as the large intestine. Now, the main function of the large intestine is basically to dry out anything that was not absorbed in your body. So any indigestible food will move on to the large intestine, and it will get dried out, and it will be eliminated from your body as feces or poop. So your large intestine extends from your ileocecal valve to the anus. There are some subdivisions. The first part of the large intestine is called the cecum. It's a small pouch at the start, so right after the chyme moves through, or any of that indigestible material moves through the ileocecal valve, it comes into the cecum. The cecum has a worm-like appendage called the appendix, and sometimes the appendix needs to be removed if it fills with bacteria and enlarges. And so sometimes we have our appendix removed. We can live without our appendix. So if you do end up with appendicitis or an infection in your appendix, it may be removed. They really don't know what the function of an appendix is. It's an appendage that hangs off of the cecum, and basically they're thinking it might have some immunity factors to it, but that's still under discussion and under research. After the cecum, it moves into the colon. The colon has 4 parts. The ascending colon moves up the right side of your abdomen. The transverse colon goes across the top of your abdomen, and the descending travels down the left side. The sigmoid is the S-shaped region that's in your pelvis that attaches to the rectum. So the colon has 4 sections: ascending, transverse, descending, and sigmoid. So after it travels through the colon, those 4 sections of the colon, it moves into a temporary storage area called the rectum. And here's where all the indigestible and non-absorbable food residues are stored until you need to get rid of it. After it fills up, after the rectum fills up, it moves into the anal canal, which then moves into the anus. There are 2 sphincters in the anus, the internal and external sphincters, and these sphincters are normally closed except when you are going to the bathroom or defecating, when you are getting rid of that waste. And so the large intestine's function is to basically dehydrate any indigestible food and deliver it to the rectum for it to be gotten rid of as waste product. One thing that you need to be aware of is there's no villi present in the large intestine. The villi, like I said, they kind of dwindle out as it gets towards the small intestine, and then once that indigestible food moves into the large intestine, there's really only cells in there that are going to absorb the water, and there's also cells that are going to put a mucus lining in the large intestine. Those are found in the mucosa and submucosa, and so they'll put that mucus lining in there so everything flows through the large intestine without too much friction. And we'll end this PowerPoint with looking at the large intestine as a whole. I want you to take note here of the yellow kind of backgroundy stuff here. That is mesentery, and that mesentery is what's holding, you can see here, holding, it's attached to the transverse colon, and it's holding to the posterior part of your body, or the back side of your body, holding it in place. If you didn't have the mesentery there, that large intestine wouldn't be suspended the way that it is in the abdomen. Okay, so food is coming in from the small intestine into the cecum. We have the appendix right here, so we're just going to go up the left of this worksheet and down the right. This is the appendix right here. So if you've had your appendix removed, that's what they were removing. The ileocecal valve, you can see is there's a cross section of the ileum with the ileocecal valve right here, and that's emptying into the cecum. And then the food is going to, well, it's really not food, it's indigestible material, is going to move up the ascending colon. It's showing you here a haustrum, like so. And then we're moving across the transverse colon, which is right here. And this bend right here is called the hepatic flexure. The reason it's called the hepatic flexure is because it's near the liver. So hepatic, anytime you see that hep prefix, that means liver. So that's why we call that the hepatic flexure. So we move across the transverse colon. We have another bend here. That is called the splenic flexure. The reason it's called splenic is, you guessed it, it is by the spleen. And we can live without our spleen. The spleen is more a circulatory organ than a digestive organ. And then we're going to go down this side called the descending colon. This S-shaped curve right here is the sigmoid colon, and the sigmoid colon empties into the rectum, into the anal canal, and then waste comes out the anus. And that is the large intestine. Hope you enjoyed that PowerPoint. We're off to accessory organs next.